Morning viewer, welcome to our Glory Globe TV as a breaking news. Viewers, before testing new therapies in animals, researchers now have a more efficient starting point. Lab-grown canine muscle cells that can help identify what works and what does not. Developed at the Texas A&M College of Veterinary Medicine and Biomedical Sciences, the Myco9 cell line is a lab-grown canine muscle cells model that allows scientists to evaluate treatments in a controlled laboratory setting before moving into further studies. The approach supports a growing national efforts to reduce animal use in research while maintaining scientific progress, particularly in fields focused on developing new therapies. Viewers, the whole purpose is to first reduce the number of animals in research and create a model that researchers can easily access and test therapies before moving into animal studies, said Dr. Peter Nagham, an associate professor in BMBS Department of Veterinary Integrative Biosciences and the principal investigator for the project. Viewers, the myoc-9 cell is line derived from canine myoblasts, precursor or primary cells that play a critical role in muscle repair and regeneration, making them an important focus for studying muscle-related disease. In the body, these cells naturally develop into muscle tissue over time. Traditionally, researchers rely on primary cells collected directly from animals or people. While useful, those cells have a limited lifespan in the lab, typically surviving only a few rounds of replications before they stop dividing and die. Viewers, when primary cells die off quickly, it limits how many treatments researchers can test and how long they can study them in the lab. Nagham said that can slow down the early stages of therapeutic development. Viewers, to overcome these limitations, researchers immortalized the myoc-9 cells by introducing a protein that allows them to replicate for beyond the normal limit. And this extended lifespan makes the cells significantly more durable and easier to work with in laboratory environment. Viewers, one of the most valuable uses of the myoc-9 cell line is in early stage therapeutic testing, helping researchers identify which treatments are all most promising before moving into animal studies. Viewers, by screening therapies in the lab fast, researchers can reduce the number of animals needed during the earliest stages of testing. Because the cells are less sensitive to handling and experimental conditions, researchers can also test therapies more consistently in the lab. So you can give them treatments like gene editing or gene therapy to see if it actually works. Nagham said, if it does, then you can move on the next phase of testing. Behind this exclusive report.